Today we are going to discuss herbal novel dosage form phytosomes. We will cover in this lecture introduction, advantages, composition and structure of phytosome, formulation of phytosomes, common stages of preparation, method of preparation, evaluation, and last one, learning objectives. Plants and its parts are widely used for the preparation of dosage form, and its chemical constituents is also used in develop the dosage form are incorporated in various dosage form. So the modern era dosage form is the phytosome. Phytosome are the novel drug delivery system. The word phyto means plant and some means cell-like. This herbal formulation is composed of bioactive phytoconstituents, mostly water-soluble compounds like flavonoids, glycoside, of herbs, extracts surrounded and bounded by lipid, as it has better absorption and bioavailability and has got the capability to cross biomembranes. The size varies from 50 nanometer to 100 micrometer. Most of the bioactive constituents of plants are polar or water soluble. However, water soluble phytoconstituents like flavonoids, tannins, glycosidal, aglycones, etc. are poorly absorbed either due to their large molecule size, which cannot be absorbed by passive diffusion, or due to their poor lipid solubility, thus severely limiting their ability to transport across lipid-rich biological membranes, resulting in their poor bioavailability. Due to poor bioavailability, the effect will show poorly. For increasing the bioavailability and its effect, the phytosome will work very nicely. The advantage of phytosomes, its water-soluble phytoconstituent can be converted into lipid compounds molecular complex. Multiple ring large size molecule cannot be absorbed by simple diffusion can be converted into phytosome. More bioavailable, better stability profile, assured delivery to the tissue. Phytosomes are used in cosmetic industries as it penetrates skin more readily and have high lipid profile. Can be taken orally or applied topically. For better illustration and understanding about the phytosome, it's known as phytomedicine. We should know composition and structure of phytosome. Phospholipids for e.g. phospholipid of natural origin. Example, phosphatidylcholine or phosphatidylserine is bifunctional. Phosphatidylcholine moiety is lipophilic. Choline moiety is hydrophilic. The hydrophilic moiety and lipophilic moiety work differently. Choline part bound with phytoconstituents forms the head, while the lipid layers forms tail. And drug moiety will entrap in the phospholipids as head and form phytosome. The drug moiety and phospholytic complex is known as phytosome. This increases the viability of the drug like terpenoids and other glycosides, which are mostly water-soluble compounds which are not viable in the body. This concept like phytosome will increase the viability in the body and increase the absorption and increase the effect of the particular drug. It means the phytosome composite with the phospholipids plus herbal extracts or drug moiety in aprotic solvent like acetone or DMSO in the ratio of 1:1 or 2:1. Phosphatidylcholine is mostly used as it has got therapeutic value in case of liver disease, alcoholic cirrhosis, drug-induced hepatitis. Phytosome is newly introduced patented technology developed to incorporate the standardized plant extract or water-soluble phytoconstituents into phospholipid to produce lipid-compatible molecular complex, which improve the absorption and bioavailability. In formulation of phytosomes, the different materials are used. First one, phospholipids, for example, phosphatidylcholine, phosphatidylcholine, aprotic solvent, dioxane, acetone, methylene chloride, non-solvent, N-hexane, and other aliphatic hydrocarbons, alcohol like ethanol and methanol. The common stages of preparation of phytosomes. First, we make the solution of phospholipids in organic solvents and entrap the drug extracts and drying in appropriate temperature and appropriate condition and appropriate manner. Then formation of thin film and then the hydration of the particular solvent and drying. So formation of phytosomal suspension. This suspension will make a clear phytosome. By formation of phytosomal suspension, after this, we have to check the entrapment of particular drug in phospholipids. It means for the checking of phytosome preparation or formation. The use of various methods for the preparation of phospholipids phytosomes. These methods can be applied for formation of phytosomes. First one, solvent evaporation method, rotatory evaporation technique, anti-solvent precipitation techniques. The method solvent evaporation. First one, we have to take drug and soya lecithin into a 100 ml round bottom flask and reflux with 20 ml acetone at the specific temperature near about 50 to 60 degrees Celsius for 2 hours, and then concentrate the mixture to 5 to 10 ml. When the mixture remain 5 to 10 ml, the precipitate will occur and this precipitate will filtered and collected. After collection of this precipitate, these are obtained as phytosome. After filtration and collection, the phytosomes obtained. The dried precipitate phytosome complex was placed in suitable bottle, especially amber-colored bottles, and stored at room temperature. The second method is rotatory evaporation technique. The specific drug and soya lecithin have to take and dissolve in 30 ml of tetrahydrofuran, stir continuously for 3 hours and maintain the specific temperature which not exceed than 40 degrees Celsius. After the complete stirring and specific time, the thin layer or film will form and then add N-hexane while stirring. After appropriate stirring, the precipitate will obtain and this precipitate will dry and pass through mess. This dried phytosome is formed. The last method is anti-solvent precipitation technique. In this technique, we have to take appropriate amount of drug with soya lecithin and reflux with 20 ml of dichloromethane at 60 degrees Celsius for 2 hours. After this, the concentrate the mixture for 5 to 10 ml and then add 20 ml of hexane. After addition of hexane, filter the precipitate formed, dry it, crush and pass through sieve number 100. This dried form is phytosome. The powder complex was placed in amber-colored glass bottle and stored at room temperature. After formation of phytosome, we have get the phytosomes. For the evaluation of the phytosome, we have to employ the different techniques which are particle size, entrapment efficacy, FTIR spectrographic analysis, differential scanning colorimetry, drug content, scanning electron microscopy, Stability study. Evaluation is needed because for the confirmation of phytosome formation and particle size, which is specific and specific criteria. And also, it is necessary for entrapment of drug with the phospholipid. These parameters are much needed for evaluation because these parameters will affect the efficacy of drug and viability of drug in the body and effect which is needed. Particle size determination can be done by using Zeta sizer Gen 3600 at fixed scattering angle of 90° at 25° Celsius. This is the specific temperature for this activity. Second one, entrapment efficacy. Loaded phytosome is diluted one-fold with 10 ml of solvent and centrifuged at 18,000 rpm or 30 minutes at -4 degrees Celsius using cooling centrifuge machine. Then supernatant is obtained and quantity of free drug determined using ultraviolet visible spectroscopy. By using this formula, entrapment of efficacy can be calculated. FTIR spectrographic analysis. In order to determine the structure and chemical stability of loaded phytosomes, phospholipids, polymer, and drug sample. Sample are crushed with potassium bromide to get pellets at 600 kg per centimeter square pressure. Spectral scanning may be done in range between 4,000 to 400 per centimeter. Differential scanning colorimetry. The drug sample, phospholipids, polymer, physical mixtures, and phytosome loaded can be placed within the aluminum creme cell and heated at 10 degrees Celsius per minute from 0 to 400 degrees Celsius in the nitrogen environment. Peak transition onset temperatures may be recorded by means of instrument. Drug content. Dissolve accurately with 100 mg of phytosome loaded in 10 ml of solvent. After appropriate dilution, absorbance may be determined by ultraviolet spectroscopy. Drug content can be calculated using this formula. Scanning electron microscopy is also known as SAME. Scanning electron microscopy used to confirm particle size distribution and surface morphology. Digital pictures may be taken by random scanning at 1,000, 5,000, 10,000, and 30,000 x magnifications. Stability study is done in order to investigate the physical appearance and leaking of the drug during storage. It is the main important condition during the storage at temperature in specific condition. These leakages can deteriorate the drug and spoil the drug. Optimized phytosomal formulation of drug extract is scaled in glass vials and stored at refrigeration temperature at 2 to 8 degrees Celsius and room temperature for a period of 3 months. Sample is withdrawn at definite time interval and observed. Stability studies are most important and most valuable for any dosage form which can be formulated. These stability studies is regulated by different authorities and it observed by the different authorities. The learning objective of this lecture is main. These are better than conventional herbal extract. They have better pharmacokinetic and pharmacological parameter. Numerous phytosomes formulations are being introduced and is profitable to the pharmaceutical, nutraceutical, or cosmetic manufacturer. They have various applications such as silimarine phytosome, phytosome of grape seeds using in anti-cancer, phytosome of green tea. It is also using in anti-cancer, antioxidants, phytosome of curcumin. Thank you.